Hi there, my name's Steve Luck, and I'm a professor at the University of California, Davis. I started learning about ERPs when I was a college student back in the 1980s, back when I had a lot more hair. I've been using ERPs ever since to study the mind and brain. Most of my research focuses on attention and working memory, both in typical individuals and in people with schizophrenia. I really like teaching other people about ERPs. For many years, I've run a series of ERP bootcamp training workshops with Dr. Emily Kappenman, a professor at San Diego State University. We also have a couple of books about ERPs. One is a general introduction to the ERP technique. The other one covers all the major ERP components. And we've put together several other resources for people who want to learn about the ERP technique. Now, these materials are primarily designed for graduate students and professors, but I also like teaching college students about ERPs. So, I created a series of lecture videos designed for students like you. Your professor wanted to use these videos in the course that you're taking, and I was happy to make the videos available. So, your course will include several of my videos, along with all the other materials that will be taught by your professor. Okay, enough background. It's time to dive in. Let's start with a completely crazy, preposterous idea, namely that it's possible to record meaningful brain activity from electrodes placed on the skin, with the skull separating the electrodes from the brain. A German psychiatrist named Hans Berger first recorded the EEG from the human scalp in the 1920s, but he was a little ahead of his time, and most neuroscientists thought he was crazy and just picking up random junk. After all, how could brain activity produced by tiny little neurons be conducted through the cortex, pass through the meninges, and cross the skull to the scalp? And yet it works. You can put electrodes on the skin overlying the skull and pick up meaningful brain activity. And it works in just about anyone, even an old thick-headed guy like me. Now, when you look at the raw EEG, it's a pretty complicated signal. It's basically the sum of everything the brain is doing. It can indicate if someone is awake or asleep, but in its raw form, the EEG can't reveal the specific neural processes that underlie perception, cognition, and emotion. However, if we apply a simple set of signal processing operations to the raw EEG, we can pull out portions of the neural activity that are related to specific sensory, cognitive, affective, and motor processes. To do this, we record the EEG while the subject performs a task. When a stimulus is presented, a marker called an event code is placed in the EEG data file, indicating what stimulus was presented and when it happened. We also have event codes for behavioral responses. A typical experiment would involve many stimuli and many responses. By using these event codes, we can process the EEG and extract the electrical potentials, the voltages, that are related to the events. That's what an event-related potential is. It's a voltage, an electrical potential, that's related to an event, like a picture on a computer screen, a spoken word, or a button press. So, what can you do with ERPs? Well, you could use them to study the development of face processing in infants and young children. You can use them to track the allocation of attention in complex scenes. You can predict which patients will wake from a coma. And you can see how anxiety and depression change perception, attention, and decision-making. You can use ERPs to understand the origins of individual differences in working memory capacity. And you can even use ERPs to decode the contents of working memory and link brain activity to computational models. Although we can do a lot with ERPs, you always need to remember that it's something of a miracle that we can record brain activity from electrodes placed on the skin with big, thick skull bones lying between the brain and the electrodes. So, there are significant limits on the kinds of information that we can extract from EEG signals. For example, we don't usually know which specific areas of the brain are producing the voltages we're recording from our scalp electrodes. But ERPs can still tell us a great deal about the mind and brain that would be difficult to learn from any other technique. I hope you find ERPs as interesting as I did when I was a student. In this series of videos, I'm going to provide a quick overview of the basics of ERP methodology so you can see the big picture. The first video will cover the basics of the EEG. To record the EEG, we put a set of electrodes in the subject's head. In most labs, the electrodes are embedded in a cap. The electrodes don't directly contact the skin. Instead, we squirt in a conductive gel that makes contact between the skin and the metal electrode pellet. But don't worry, that's a blunt-tip syringe, not a sharp needle. We don't inject anything into the skin. After we squirt in the gel, we connect each electrode to an amplifier, which then feeds into a computer so we can see the EEG on the screen and record it to the computer's hard drive. Here we're looking at a 5-second period of EEG, starting 50 seconds into the recording. We get a separate signal at each electrode site. The x-axis is time. Here it's shown in seconds, but we often use milliseconds. There are 1,000 milliseconds in 1 second. The y-axis is voltage, usually expressed in microvolts, where 1 microvolt is 1 millionth of a volt. Notice the oscillations at these electrode sites here. If you counted, you'd find about 10 peaks in a 1 second period. That means it's a 10 Hz oscillation. Oscillations around 10 Hz are really common in EEG recordings. They were actually the first feature of the EEG that Hans Berger documented back in the 1920s. He called them alpha band oscillations. Over time, researchers discovered that the EEG contains oscillations in several different frequency bands, which are known as the alpha, beta, gamma, theta, and delta bands. You mainly see delta band oscillations when someone is asleep. In fact, these are the slow waves that give slow wave sleep its name. Theta band oscillations are prominent in microelectrode recordings from the hippocampus in animals, but you can't see hippocampal theta on the scalp. However, you can see theta band oscillations that are generated in neocortical areas, such as the prefrontal cortex. Alpha band oscillations are mainly seen over visual cortex when the subject is awake but zoned out, focusing on their own internal thoughts rather than on an external task. Most of the ERP experiments in my lab are pretty boring, so we see a lot of alpha. Alpha band oscillations are usually thought to reflect corticothalamic feedback loops. Beta band oscillations typically occur when someone is mentally active, often when there's very little alpha. The beta band oscillations tend to be much smaller and less regular than the alpha band oscillations. At the upper end, we see gamma band oscillations. They occur so rapidly that they presumably reflect short-range feedback loops within a cortical column. Gamma band oscillations tend to be quite small, and they can be difficult to distinguish from muscle artifacts in scalp recordings. I'd like to caution you that this is just a coarse way of subdividing the EEG. You don't want to assume that the theta band activity you see in one experimental paradigm represents the same underlying neurocognitive processes as the theta band activity you see in a very different paradigm. However, these 5 frequency bands, delta, theta, alpha, beta, and gamma, have proven to be a useful first approximation for dividing up the oscillations in the EEG. Now let's talk about how we extract ERPs from the raw EEG using signal averaging. As I mentioned before, the EEG is basically the sum of everything the brain is doing. It can indicate someone's general state, whether they're asleep, drowsy, or wide awake, but the raw EEG can't reveal the detailed neurocognitive processes that underlie thought and emotion. For that, we want to look at event-related potentials, voltages that are related to a specific event, such as a stimulus or a response. And to see the ERPs, we need to know exactly when each event happened. So most labs use 2 computers, one that presents the stimuli and records the responses, and another that records the EEG. The stimuli are usually auditory or visual, but some studies use somatosensory, olfactory, or even gustatory stimuli. Anytime a stimulus is presented or a response occurs, an event code is sent from the stimulus presentation computer to the recording computer. This indicates what the event was and exactly when it occurred. If we look at the raw EEG, there isn't a very consistent response following each stimulus. That's because the brain's doing a million other things in addition to processing our little stimulus. To pull out the brain's response to the stimulus, we use a very simple procedure called signal averaging. First, we grab the period of EEG following each stimulus. This is called segmenting the EEG. Each segment is called an EEG epoch. Now we can take all the EEG epochs and line them up in time. In this example, each epoch contains 600 milliseconds of EEG following the stimulus onset. The epoch length depends on the nature of the specific experiment, but it's usually somewhere between 10 milliseconds and 2000 milliseconds. To pull out the brain's consistent response to some type of event, we can simply average across the epochs for that event type. When we average across enough epochs, any activity that's consistent from trial to trial remains in the average, and any random noise simply averages out. This gives us an averaged ERP waveform. Each time point in the average waveform is simply the mean across epochs at that time point. In a typical ERP experiment, we time lock to the onset of a stimulus. In other words, time 0 is stimulus onset. When we do this, we can track the flow of information about the stimulus through the brain over a period of several hundred milliseconds. For a visual stimulus, we initially see the C1 wave, which comes from primary visual cortex. Then we see this P1 wave, which comes from higher level visual areas. We then see activity related to stimulus categorization, response selection, and working memory encoding. For stimuli with a strong emotional content, we might also see emotion-related activity. As we'll discuss in another video, there's no delay between when the brain activity happens and when the voltage is picked up by our scalp electrodes. So a voltage at 293 milliseconds reflects brain activity that happened exactly at 293 milliseconds. So ERPs allow us to see the flow of information through the brain, millisecond by millisecond. In other words, the ERP technique has great temporal resolution. And given how quickly the brain works, this amazing temporal resolution allows us to study important aspects of brain function that are impossible to pull apart with techniques like fMRI that depend on the sluggish hemodynamic response. However, the ERP technique has poor spatial resolution. If you want to know where in the brain something happened, fMRI is usually much better. Although most researchers focus on stimulus-locked averages, where time zero is stimulus onset, you can instead time lock to the response. This gives you a response-locked average. In this example, the mean response time was 576 milliseconds. That's 576 milliseconds after the time locking point in the stimulus-locked average. In the response-locked average, this means that the stimulus was 576 milliseconds before the time locking point. If the response time were 570 milliseconds on every trial, the response-locked average would be identical to the stimulus-locked average, but shifted over by 576 milliseconds. However, there's a lot of trial-by-trial variability in response time. This variability smears out the response-related activity in the stimulus-locked average, and it smears out the stimulus-related activity in the response-locked average. So it can be useful to look at both stimulus-locked and response-locked averages. In this video, I'm going to provide a quick example of an actual ERP study so that you can see how ERPs are used to answer interesting questions about the operation of the mind and brain. I'll be talking about this study by Jim Tanaka and Tim Curran. I'm going to gloss over the details, so I encourage you to read the paper. It's a great paper for both beginners and experts. It focuses on the N170 component, which is widely used to study face perception. Here are the stimuli from a typical N170 experiment. Subjects were shown a sequence of faces and cars, each of which was flashed briefly on a computer screen. For each subject, a face ERP was computed by averaging across all the faces, and a car ERP was computed by averaging across all the cars. The waveforms shown here are averaged across all the subjects. We call these grand average waveforms. As you can see, the N170 wave, the negative deflection that peaks at around 170 milliseconds after stimulus onset, was much bigger for the faces than for the cars. Many studies have found similar results. Faces generate a larger N170 than just about any other stimulus category. Of course, fMRI studies have also found that faces generate larger bold responses than most other stimuli in certain brain areas. This research has led to a fundamental theoretical question. Is face perception special? That is, do we have domain-specific neural systems that are solely used for face perception? That might explain why inverting an image has a larger impact on the perceptibility of faces than on other sorts of objects. But maybe these effects are a consequence of the fact that virtually all humans have a lifetime of experience in perceiving faces. In other words, are there special purpose processes for faces, or do we use the same processes for all stimuli that we're experts at perceiving? This is the question that Tanaka and Curran wanted to answer using the N170 component. Specifically, they wanted to know whether the N170 is impacted by perceptual expertise. To answer this question, they recruited people with different kinds of perceptual expertise. They found 15 bird experts and 15 dog experts, and then they showed these people pictures of birds and pictures of dogs. They used a 2x2 factorial design with a between subjects factor of expertise type and a within subjects factor of stimulus type. On each trial, subjects saw a category name followed by a picture of a bird or a picture of a dog. At the end of the trial, subjects had to indicate whether or not the picture they just saw was a member of the specified category for that trial. But the specific task doesn't usually have much impact on the N170. If the N170 component is influenced by expertise, then they predicted they would see a larger N170 for birds than for dogs in bird experts, but a larger N170 for dogs than for birds in dog experts, and that's exactly what they found. You can see that the N170 was larger for bird pictures than for dog pictures in the bird experts. But the N170 was larger for dog pictures than for bird pictures in the dog experts. So just as almost everyone is highly experienced with face processing, and almost everyone has a larger N170 for faces than for other kinds of stimuli, bird and dog experts have a larger N170 for stimuli in their domain of expertise. This is consistent with the idea that face perception is achieved by a set of general-purpose processes that aren't face-specific but depend on expertise. Now, one experiment isn't enough to settle this complex issue, but this study certainly provides useful evidence. In this video, we're going to talk about how we deal with noise when trying to extract the ERP signal from the EEG. By noise, I don't mean sounds, I mean random fluctuations in the EEG. We use signal averaging to deal with these random fluctuations. The more trials we have, the more random activity will average out, leaving us with a beautiful ERP waveform. But this leads to an important question. How many trials do you need to average together? Well, most questions like this have what I like to call a two-word answer that begins with it and ends with depends. In this case, it depends on the size of the signal relative to the size of the noise, the signal-to-noise ratio. Depending on the experiment, we might need 10 trials or we might need 1,000 trials to get an acceptable signal-to-noise ratio. Here's an example where we have only 8 trials, which isn't even close to enough in most experiments. This experiment used an oddball paradigm in which subjects pressed one of 2 buttons on each trial to indicate whether the stimulus was an X or an O. The O's were rare, so we call those the oddball stimuli. Here we're showing the EEG epochs from 8 oddball trials. You can see a lot of variation from trial to trial, but all 8 trials have a positive voltage from approximately 300 to 600 milliseconds. That's the P3 wave, which is a very large positive component that you get for oddballs in this paradigm. Here you can see the average of the 8 oddball epochs. We actually had way more than 8 oddballs, but I wanted you to see what it looks like if you have a small number of trials. You can see the P3 wave, a broad positivity from approximately 300 to 600 milliseconds. In this particular example, the P3 is huge, about 25 microvolts. It's way bigger than the noise in the pre-stimulus interval. But this is an unusual situation. Usually an ERP component is only a few microvolts, and we're looking for differences between conditions that might be less than 1 microvolt. So we usually need way more than 8 trials. For example, this P2 wave isn't any bigger than the noise in the pre-stimulus period. If the signal isn't any bigger than the noise, it's going to be hard to find statistically significant differences between conditions or groups. Now, if we make some simple assumptions, it turns out that the signal-to-noise ratio will increase in proportion to the square root of the number of trials. So doubling the number of trials from 8 to 16 would only increase the signal-to-noise ratio by 41%, because 1.41 is the square root of 2. We'd need to quadruple the number of trials to double the signal-to-noise ratio, because the square root of 4 is 2. So we'd need 32 trials to double the SNR that we get with 8 trials. Increasing the number of trials doesn't change the signal, it just decreases the noise. So if we quadruple the number of trials to double the SNR, this would effectively cut the noise in half. The square root rule can be a major source of frustration when you're designing an ERP experiment. You can only quadruple the number of trials so many times before the experiment becomes impractical. As a result, the ERP technique is usually limited to paradigms where you can get at least a few dozen trials from each subject in each condition. Given that averaging is used to reduce the noise in the ERP waveform, and it's often difficult to get enough trials to produce an acceptable signal-to-noise ratio, it's important to ask where the noise comes from. There are 3 main noise sources. First, the EEG contains a ton of genuine brain activity that just isn't time-locked to the stimulus. For example, you can see alpha band oscillations here during the pre-stimulus interval. Because they're present before the stimulus appears, they're clearly not triggered by the stimulus, so they add uncontrolled variability to our average ERPs. Biological artifacts that are generated outside the brain are the second major source of noise in EEG recordings. For example, eye blinks and eye movements create large voltages called the electrooculogram. These voltages conduct to our scalp electrodes, often creating artifacts of over 100 microvolts. The muscles on the head and neck also create high-frequency voltages called the electromyogram that can contaminate the EEG. And the skin itself is an electric organ that produces slow voltage changes called skin potentials. The third source of noise in EEG recordings is induced electrical activity from the recording environment. When a current flows through a conductor, it's accompanied by a magnetic field. If that magnetic field then passes through another conductor, it induces the flow of electric current in that second conductor. As a result, AC electrical current flowing back and forth through a light bulb at 60 Hz will create a 60 Hz magnetic field oscillation, which then induces a 60 Hz voltage oscillation in our EEG electrodes. Or it may be 50 Hz, depending on what country you're in. Now let's ask a fundamental question. What do we actually mean by noise? Generally, noise just means any source of uncontrolled variability in the recording. When this variability comes from light bulbs or the skin, it's pretty straightforward to see what we mean by noise. But what about these alpha oscillations? They reflect real brain activity, and they can be used to study things like attention and mind wandering. So aren't these oscillations a signal rather than a source of noise? Well, one researcher's signal can be another researcher's noise. The key is whether it creates uncontrolled variability in the signal of interest. If we're trying to study the processing of the stimulus, we typically have no control over the alpha band oscillations, and they create uncontrolled variability in the stimulus-locked ERP waveform. That's noise. There's also trial-by-trial variability in the actual neural response to the stimulus. If we don't experimentally control this variability, then it's considered noise. Keep in mind that averaging is a pretty simplistic way of dealing with noise. ERP researchers also use a variety of more sophisticated ways of dealing with things like alpha band oscillations and trial-by-trial variations in ERP responses. But these methods have their limitations as well, so averaging is still the most common way of dealing with noise. Now we're going to take a look at some common conventions in ERP research, so you understand what you're looking at when you see published data. First, let's consider the most common system for naming electrode locations, called the International 1020 system. It's called the 1020 system because it involves measuring the head and placing electrodes every 10 or 20% of the distance between key landmarks. This system was developed in 1958, before it was possible to record from large numbers of electrodes, so now we might place the electrodes at 5 or 10% distances. Each electrode name begins with one or two letters indicating the general region of the brain, such as FP for frontal pole or O for occipital. These letters can be combined for in-between regions, such as PO for electrodes near the parietal-occipital border. The letters are followed by a number. We use odd numbers for the left hemisphere, even numbers for the right hemisphere, and a Z to indicate 0 for the midline. The numbers get larger as we move farther away from the midline. For example, as we move away from the midline toward the left side over frontal cortex, we have electrodes F1, F3, F5, F7, and F9. Now that you know the general system, you don't need to memorize every individual electrode name. If I ask you where electrode FC2 is located, you should be able to figure out that it's near the border of the frontal and central areas, in the right hemisphere, and close to the midline. Some EEG recording systems don't use the 1020 system. Instead, they use a geodesic configuration that guarantees an equal spacing between each adjacent pair of electrodes. These layouts use idiosyncratic numbering systems, and a publication will typically say something like, "These are data from electrode 27, which is near the P2 location in the International 1020 system." Now let's talk about conventions for plotting ERP data. You might have noticed that the ERP waveforms so far have shown only a single electrode, even though most labs typically record from between 20 and 128 electrodes. If we showed the data from every electrode site, you'd end up with something like this, where it's nearly impossible to see what's going on. We record from many electrode sites simultaneously, but the waveforms at nearby sites are typically quite similar, so there's usually no point in showing all of the electrode sites in a journal article. Instead, we typically show the data from a single electrode site, or maybe a few sites, and then we show scalp maps to show the distribution of voltage at a given moment in time. Note that these maps use interpolation to show voltage over the entire scalp, even though we only have measures at the discrete electrode sites. So you can see gradual variations in shading between the electrode sites, which reflect interpolated voltage values. That just makes the maps easier to look at, especially if you're used to looking at fMRI data. Another thing you should be aware of is that ERP papers don't usually show single subject ERP waveforms. Again, this is largely to avoid overwhelming the reader with a million squiggly lines. Here we have the average ERPs from only 8 subjects in a single condition, and it's already a lot of information. Instead, we usually take the single subject average ERP waveforms and average them together into what we call a grand average waveform. So when you see ERP waveforms in a paper like this, you're looking at grand averages across the subjects. However, the statistical analysis is done using the single subject waveforms, not the grand averages. For example, this study measured the mean amplitude in the N170 time range for each individual subject's average ERP waveform in each condition, and then put those single subject amplitude values into a simple analysis of variance. Now I'd like to briefly discuss an oddity about how ERPs are sometimes plotted. Here's a figure from the first edition of my ERP book, and here's the same figure from the second edition. Do you see the difference? In the first edition, all the figures were plotted with negative upward. That's how I was trained to do it back in the 1980s. This odd convention was a result of the nature of the hardware used by early researchers to record EEG and ERPs. These researchers didn't have computers, and it would have been very difficult for them to plot positive upward given the hardware they were using. But this tradition persisted long after computers made it trivial to plot the data with positive upward, like the other 99% of scientists did. I eventually saw the light, so in the second edition of my book, all the figures were plotted with positive upward. The bottom line is that plotting negative up is an outdated convention, but it's still pretty common, so you need to make sure you look at the polarity indicators in the plots of ERP waveforms. In this next set of videos, I'm going to talk about ERP components, what they are, where they come from, and what they mean. In this first video in the series, I'm going to provide a brief overview of what we mean by the term ERP component and how the components are related to the peaks we see in the waveforms. The terms peak and component are sometimes used synonymously, but they really refer to different things. Peaks are things that we can observe in our scalp recordings, whereas components occur in the brain and can't be directly observed from scalp electrodes. The observed peaks are the result of the underlying brain components, but the relationship can be complex. So be careful. Peaks and components are not the same thing. And one of the most challenging things about ERP research is to go beyond the observed peaks and understand what's happening with the underlying components. Let's start by talking about how ERPs are generated. This will be a simplified description. I cover ERP generation in more detail in another set of videos. With a few rare exceptions, ERPs are generated by cortical pyramidal cells during neurotransmission. To be visible on the scalp, a large number of neurons must be active at the same time. Typically, this would be a cluster of neurons in some functionally defined area of the cortex. The extracellular electric fields produced during neurotransmission sum across the neurons in an area, forming what we call an equivalent current dipole that points perpendicular to the active cortical surface. This will give us a negativity in our scalp EEG electrodes on one side of the dipole and a positivity on the other side. If you'd like more details, here are a couple of excellent review papers. An ERP component is essentially the extracellular voltage produced by a cluster of functionally related neurons in the brain. The sequence of positive and negative peaks that we see at a given scalp electrode reflects the sum of many of these components, each of which has its own time course. The components overlap in time, and it's hard to tell when a single component actually begins and ends by looking at the combined contribution of multiple components that we record from our scalp electrodes. For example, imagine that we have the 3 components labeled here as C1, C2, and C3. Each internal component is represented by an arrow, showing the location and direction of the dipole for that component. And each component also has what we call a source waveform, which is the change in voltage over time in response to a given event at the source. And imagine that we have the 3 electrodes shown here, labeled E1, E2, and E3. To understand how the underlying components combine together in those scalp electrodes, you need to know a key fact about ERPs. Voltages simply sum together in a conductor. As a result, the voltage recorded at each electrode will be a weighted sum of the underlying source waveforms. There's a different weighting between each component and a given electrode, reflecting the location and orientation of the dipole, the location of the electrode, and the conductivity of the brain, meninges, skull, and scalp. For example, there's one weight between component 1 and electrode 1, and another weight between component 2 and electrode 1, and a third weight between component 3 and electrode 1. You have a different weight for each combination of component and electrode site. The contribution of a given source to a given electrode site at a given time is simply the amplitude of the source waveform at that time multiplied by the weight between that component and the electrode site. Some of the weights may be negative, which just means that the electrode is on the opposite side of the dipole. In this example, there's a negative weight from component 2 to electrode 3, so even though the source waveform is negative going, you see a positive going deflection at that time in the scalp voltage. Any given electrode picks up a large number of different components. In this example, all 3 components contribute significantly to the voltage at all 3 electrodes. Here's another example. You can see that we have 3 components and 3 peaks. But notice that the first component peaks at 100 milliseconds, whereas the first peak in the scalp waveform is at 50 milliseconds. And it looks like the second peak goes from about 90 to 180 milliseconds, but the underlying component actually goes from about 50 to 225 milliseconds. And there are usually way more components than there are obvious peaks. For example, there are at least 10 distinct components active between 50 and 150 milliseconds in the sensory response to a visual stimulus. And there are clearly several different N2 components that come from different brain areas and reflect fundamentally different neural computations. So, peaks and components are not the same thing. We see the peaks, but we can only draw indirect inferences about the underlying components. If you'd like to know more about how ERP components are defined and how they can be estimated from the observed scalp waveforms, take a look at this chapter that Emily and I wrote for the Oxford Handbook of ERP Components. Now I'd like to turn to the conventions used to name peaks and components. In many cases, people use the same name for the observed peak and the underlying component, which muddies the distinction between peaks and components. The most common convention is to label the peaks with a P or N to indicate positive or negative, followed by a number, like the P3 wave shown here. In some cases, the number indicates the ordinal position in the waveform. For example, P1, P2, and P3 are the first, second, and third major positive components. In other cases, the number indicates the latency in milliseconds. For example, the visual N1 often peaks around 170 milliseconds, so it would be called N170. If the number is less than 5, you can assume it's the ordinal position. If it's greater than 5, you can assume it's the latency in milliseconds. The P or N doesn't indicate whether the voltage is positive or negative per se. For example, the voltage for this N2 wave is on the positive side of the zero line. Instead, the P or N indicates whether the peak is heading in a positive or negative direction. The N2 here is negative going, so we give it an N. The idea is that there's actually a negative component in the brain, but the voltage from this component is superimposed on positive voltages from other components at this particular electrode site, so the scalp voltage never quite becomes negative. This naming convention might seem straightforward, but it can get pretty complicated and confusing. As I mentioned before, the relationship between the timing of the underlying component and the timing of the observed peak can be pretty complicated. And there are way more underlying components than visible peaks. In fact, a given component might not have a single distinct peak. Also, every component produces a positive voltage over one side of the head and a negative voltage over the other side. We might not have electrodes covering enough of the head to see both sides of the dipole, but both the positive and negative sides of a component are present somewhere on the head. So a component could be a P1 at one electrode and an N1 at another. We don't even use P or N for the very first visual response, which comes from primary visual cortex. We call this response C1 because it can be either positive or negative depending on whether the stimulus is presented above or below the point of fixation. This is because primary visual cortex is folded up in the calcarine fissure, and the upper and lower visual fields project to opposite sides of the fissure. This flips the polarity of the dipole for stimuli in the lower field relative to stimuli in the upper field, giving us opposite C1 polarities at our scalp electrode. The number in a component label can also be confusing. The P3 wave is often called P300 because it peaked at around 300 milliseconds in the first study where it was observed. But it can peak as late as 600 milliseconds in some experiments. And yet it's often called P300 under the assumption that it's the same brain process but occurs at different times depending on the difficulty of the task. You should also know that some components are modality specific and others are modality independent. For example, the visual N1 is completely unrelated to the auditory N1, but you get the same P3 wave for both auditory and visual stimuli. The bottom line is that the nomenclature for naming peaks is a bit of a mess. It's kind of like the English language, where a given word can have multiple meanings, and different words can have the same meaning. We deal with this kind of ambiguity in natural language comprehension by using the context to figure out what a word means. And once you become fluent in the language of ERPs, you'll also be able to use the context to know what somebody means when they refer to a component like P600. In the meantime, try not to read too much into the label that somebody uses for an ERP component. The fact that two different papers refer to the N1 wave doesn't mean that they're actually talking about the same component. Now I'm going to provide a quick overview of some common ERP components to give you an idea of some of the general topics that can be studied with ERPs. We'll start with sensory components and then move on to later components that reflect higher-level cognitive and affective processes. I've already shown you this artificial ERP waveform to convey the general idea that the sequence of voltages over time reflects the sequence of processes that are triggered by a stimulus. Now we're going to dive a little deeper into visual sensory responses. We've already talked a little about the C1 wave, which comes from primary visual cortex and can be either positive or negative depending on whether the stimulus is in the lower or upper visual field. When it's positive, it merges together with the P1, so you don't usually see a distinct C1 unless the stimulus is in the upper visual field and the C1 is negative. The P1 and N1 waves reflect the combination of many different brain areas, most of which are in higher level areas of visual cortex. As you might expect for electrophysiological signatures of sensory processing, the C1, P1, and N1 waves are highly sensitive to the physical properties of the stimulus, such as brightness. And as discussed in another video, the N1 or N170 wave is bigger for faces than for most other classes of stimuli. Experience also plays a role. For example, words elicit a large N1 in experienced readers. After decades of ERP research, I bet I have a huge N1 when I look at ERP waveforms. The P1 and N1 are larger for attended location stimuli than for ignored location stimuli. However, these effects are typically observed only for spatial attention and only when attention has shifted prior to stimulus onset. Other kinds of attention don't usually have an impact until later components. For example, we see a component called N2PC if attention shifts to a location after stimulus onset. Auditory stimuli generate a very different pattern of sensory ERP responses. Whereas the first visual ERPs start around 50 milliseconds, you can see auditory ERPs within about a millisecond. The auditory system is way faster than the visual system. The initial auditory ERPs come from the brainstem and are called the auditory brainstem responses, or ABRs. They're the one common exception to the rule that ERPs are ordinarily generated by cortical pyramidal cells. Note that the ABRs are labeled with Roman numerals. They're used to diagnose hearing impairments in clinical audiology, and they're often measured in newborn infants to screen for hearing problems. Both of my kids were screened in the hospital shortly after they were born. I really got a kick out of that. If we look on a longer time scale and use different filter settings, we can see the mid-latency auditory responses, which are generated in the medial geniculate nucleus and the primary auditory cortex. On an even longer time scale, we can see what are called the long-latency auditory responses, which come from a variety of cortical areas. Another interesting auditory component is the mismatch negativity. We see this component if we use an auditory oddball paradigm. For example, we might present a tone every 500 milliseconds, with 80% being 1000 Hz standard tones and the other 20% being 1500 Hz deviant tones. It sounds like this. This would keep going for several minutes. The deviant tones elicit a larger negative response than the standards, around 200 milliseconds. This is called the mismatch negativity, or MMN. You can get an MMN with virtually any type of physical deviance that someone can perceive. The data shown here are from the ERP core MMN task, where we used intensity deviance. The MMN doesn't require that the subject's performing a task. It's an automatic response to the mismatch. In fact, many researchers record the MMN while subjects read a book. In the ERP core, we had subjects watch this silent movie while the tones were playing. The fact that you don't need a task makes the MMN well suited for use in infants, where it's been used to study the development of phoneme discrimination. Instead of using different pitches, these studies use different phonemes, like da, 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 ta, da, 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 ta, da. The MMN is also useful in a variety of patient groups. For example, it can be used to predict which coma patients will recover. It's also widely used in schizophrenia research. Because there's no task, you don't have to worry about differential task performance as a confound. You can read more about the mismatch negativity in this chapter by Naughton and Kriegapoo in the Oxford Handbook of ERP Components. Now we're going to talk about some of the ERP components that reflect task-dependent cognitive processes. We'll start with an attention-related component called N2PC. Imagine that you'd like to eat a banana. Before your eyes move to the banana in this fruit bowl, you use covert attentional processes to find the banana and help you plan the eye movement. The term covert attention refers to a shift of internal processing resources, whereas we use the term overt attention when the eyes actually move to the object. An ERP component called N2PC can be used to track the covert focusing of spatial attention. The PC in N2PC stands for posterior contralateral. As you probably learned at some point, the sensory and motor systems have a contralateral organization, with the left hemisphere coding the right side of space and the right hemisphere coding the left side of space. The N2PC is a negative going voltage over posterior electrode sites contralateral to the object being attended. If you're covertly attending to the banana while looking at the center of the fruit bowl, the banana is mainly projecting to the right hemisphere, which is the contralateral hemisphere, or you'd see the N2PC component over the right hemisphere. Of course, most experiments use much more controlled stimuli like these. In this task, subjects attend to the red square in some trial blocks and to the green square in others. The task is to report whether the gap in the attended square is on the bottom or on the top. The stimulus locations are randomized from trial to trial, so when the display appears, the subject has to search for the target. We're studying covert attention here, so we have subjects keep their eyes locked on the central fixation point and use their peripheral vision to do the task. If we look over the left hemisphere, we see that the P1 and N1 components are the same whether the target is in the left visual field or the right visual field. P1 and N1 are only impacted by attention when attention is focused on the location of the target before the stimulus array appears. Once the stimuli appear, it takes about 200 milliseconds for attention to shift to the target, and then we'll see the N2PC as a negative going wave over the contralateral hemisphere. For the left hemisphere, that means we see a more negative voltage from about 200 to 300 milliseconds when the target is on the right side than when it's on the left side. But the N2PC is added onto all the other ERP components that are active during this time period, so the overall voltage during this period is actually positive. The N2PC is a negative deflection that's added onto this positive voltage. If we look at the right hemisphere, we see the complementary pattern. Now the voltage is more negative for a left visual field target than for a right visual field target. We can simplify this by collapsing over the left and right hemispheres. The contralateral waveform is the average of the right hemisphere for the left target and the left hemisphere for the right target. The ipsilateral waveform is the average of the left hemisphere for the left target and the right hemisphere for the right target. The N2PC is defined as the difference in voltage between the contralateral and ipsilateral waveforms. The N2PC allows us to see shifts of covert attention. In other words, we can measure a mental operation that occurs entirely in the head and can't be directly observed in behavior. And that's one of the main virtues of the ERP technique. It allows us to observe the neurocognitive processes that lead to behavior, but cannot be directly observed in behavior. Here's an example from a study that Emily did several years ago. Subjects saw a threat image paired with a neutral image. The stimuli were task irrelevant, and the threat image was equally likely to be on the left side and on the right side. Different images were presented on different trials. Many theories predict that covert attention will be automatically attracted to the threat image, but most behavioral studies using indirect measures of covert attention don't actually see any capture of attention by the threat image. But we were able to directly measure the allocation of attention using the N2PC component. As you can see, Emily found that the threat image elicited a robust N2PC. The voltage was more negative contralateral to the threat than ipsilateral to the threat. Tons of other studies have also used the N2PC component to track the allocation of attention to other kinds of stimuli. Attention is closely related to working memory, and it's possible to take our N2PC attention paradigm and modify it to see an ERP component related to working memory called contralateral delay activity, or CDA. In this task, subjects are asked to remember the colors of the circles and ignore the rectangles. They only have to remember the colors for about a second, and then they see a test array. They then indicate whether the colors of any of the circles have changed. In this example, the red circle changed to blue. The rectangles never change color. In this kind of paradigm, you initially see an N2PC component as attention is shifted to the circles. This is followed by the CDA, or contralateral delay activity, a sustained voltage over the hemisphere contralateral to the objects being maintained in memory. The N2PC and CDA are added on to other components, but you can isolate them by looking at the difference between the contralateral and ipsilateral waveforms. Although they're both contralateral negativities, the N2PC and CDA have different scalp distributions, so they're distinct components. The N2PC reflects the initial focusing of attention onto a visible stimulus, and the CDA represents the maintenance of information in working memory after the stimulus has disappeared. One of the coolest things about the CDA is that it's strongly correlated with individual differences in behaviorally measured working memory capacity. It's not often that you can see such a strong correlation between brain activity and individual differences in cognitive ability. If you'd like to know more about ERP components related to attention and working memory, I recommend these 3 chapters in the Oxford Handbook of ERP Components. Here's an amazing new direction in studying attention and working memory with ERPs. We can use machine learning algorithms to decode the contents of working memory on the basis of the pattern of voltage over the scalp. For example, in this study, Gil Bey presented a series of faces drawn from a set of 16 combinations of 4 identities and 4 expressions. Subjects simply had to remember the most recent face, and their memory was tested after a random 12% of faces. Gil then trained a set of support vector machines to classify the identity of the face independently of the expression. He trained another set of support vector machines to classify the expression independently of the identity. The ERP scalp distributions differ only slightly among the 4 identities and 4 faces, but the support vector machines could pull out these subtle differences. As a result, decoding accuracy was well above chance, 25%, for both identity and expression. Moreover, the decoding was done separately at each time point so that Gil could determine what information was present in the signal at each moment in time, first during the stimulus and then during the delay period. Although the decoding was more accurate when the stimulus was visible, information about both identity and expression could be decoded from working memory after the face disappeared. Pretty amazing, huh? It's especially amazing given that Gil was recording brain activity from electrodes on the skin outside of the skull. The decoding accuracy never got above around 40% correct, and the decoding was done on average ERPs rather than single trials. So, the scalp signal doesn't have enough information about faces to be used for engineering applications like brain-computer interfaces. We can't realistically use this approach to read your mind, but maybe that's a good thing. Now let's talk about ERP components related to language. You can read about these components in the chapter that Tamara Swab's lab wrote for the Oxford Handbook of ERP Components. The most famous language-related ERP component is the N400. Here's an example of a classic N400 paradigm, which was originally developed by Marta Kudas and Steve Hilliard. In this paradigm, sentences are presented visually, one word at a time, while the EEG is being recorded. Subjects just read the sentences. For example, the subject might see a sentence like, "I take my coffee with cream and sugar." This is the ERP waveform elicited by the word sugar at the end of the sentence. Time 0 is the onset of the final word in the sentence, and we're looking at the brain's response to that word. Of course, these are artificial data, not the real thing. We might also have a sentence like the one shown at the bottom of the screen here. "I take my coffee with cream and dog." If the meaning of the last word doesn't fit well with the rest of the sentence, you get a very different brain response. The initial sensory processing is the same for the 2 words, but the semantically incongruous word elicits this big N400 wave, peaking around 400 milliseconds after the onset of the word. The N400 appears to reflect the work your brain is doing as it tries to figure out what the word is and how it fits into the sentence. If more work is needed, you get a larger N400. And the size of the N400 for a given word reflects the extent to which that word can be predicted from the preceding context. As a result, you don't need bizarre sentences like, "I take my coffee with cream and dog" to see the N400. For example, "I placed my keys on the kitchen floor" is a perfectly reasonable thing to say, but floor is somewhat unexpected, so it would give you a medium-sized N400. If the sentence ended in table, that would be more expected, so table would elicit only a small N400. Words can also elicit an N400 even if they don't occur in a sentence. For example, in a semantic priming experiment, nurse would elicit a large N400 if preceded by an unrelated word like tree, but not if preceded by a related word like doctor. These effects can also be seen when people listen to natural speech rather than reading words on a computer monitor. The general idea is that every word you see or hear generates some amount of N400, with a larger amplitude to the extent that the word can't be predicted from what came before it. For example, in this famous quote from John Lennon, the first word is completely unpredictable and would generate a large N400. The next word, is a little more predictable and would generate a smaller N400. By the time you've read life is what happens, when is pretty predictable and would generate a smallish N400. And by the time you get to the end of the sentence, plans is highly predictable and would generate very little N400. But keep in mind that the N400 is about the predictability of the meaning, not about syntactic predictability. If we make the sentence grammatically incorrect by changing making into make, you wouldn't get an N400 to make. Instead, you'd get a different component called P600. Now we're going to shift to two related ERP components, the P3 wave and the late positive potential. The P3 or P300 is present in almost any task, but it's classically observed in the oddball paradigm. We could take the auditory oddball paradigm we previously discussed for the mismatch negativity, slow it down a little, and have subjects press a button for each stimulus to indicate whether it's the lower pitch or the higher pitch. It would sound like this. In paradigms like this, the oddball generates a much larger P3 than the standard. The P3 is largest near the PZ electrode site, but it can be seen at virtually every electrode site on the top half of the head. You can get this kind of effect for virtually any kind of stimuli, as long as the task requires the subject to classify the stimuli into a frequent category and a rare category. And it's the task-defined categories that matter, not the probabilities of the physical stimuli per se. For example, imagine that we present the letters A, B, C, D, and E with equal probability in random order on a video monitor. And imagine that we ask subjects to press one button for the letter B and another button for any other letter. This task requires classifying B as one category with a probability of 20%. And it requires grouping the other four letters together as a separate category with a probability of 80%. Although each individual non-target letter has a probability of 20%, they're grouped into a task-defined category with a probability of 80%. We should therefore get a bigger P3 for B's than for the other letters. This is how the P3 task in the ERP core works, except that B wasn't always the target letter. A different letter was defined as the oddball for each block of trials. As you can see, the oddball generated a larger P3 than the standards, because the probability of the standard category was 80%, even though each individual standard letter had a probability of 20%. In general, the amplitude of the P3 is inversely related to the probability of the stimulus. If we use probabilities of 10% and 90% rather than 20% and 80%, we'd get an even bigger P3 for the oddballs and an even smaller P3 for the standards. I could talk about the P3 for hours, but instead have a look at this chapter by John Polich in the Oxford Handbook of ERP Components. Now let's shift from cognition to emotion, which is summarized in the chapter from Greg Hychek's lab. There are several emotion-related ERP components, but here I'll focus on the late positive potential, or LPP. As you probably know, emotion has two main dimensions, arousal and valence. Arousal is the intensity of the emotional response, and valence is whether it's positive or negative. The LPP primarily reflects arousal and not valence. In this study, people looked at pleasant pictures like babies laughing, unpleasant pictures like rotting meat, and neutral pictures like desks. Both the pleasant and unpleasant pictures were high in arousal, so both elicited a large LPP. Some researchers think that the LPP is the same thing as the P3. It has a pretty similar scalp distribution. But other researchers think that other sources also contribute to the LPP. There are lots of other interesting ERP components as well. I don't have time to go into all of them now, but you can read about them, guess where, in the Oxford Handbook of ERP Components. In this next set of videos, I'm going to dive a little